For this next video in Unit 4, we're going to be talking about cerebrospinal fluid, and I think the other one is synovial fluid. So the functions of CSF. So it supplies nutrients to the nervous tissue, it removes metabolic wastes, and it provides a cushion for the brain and spinal cord to protect against trauma. So the brain and spinal cord are lined by the meninges, and in the meninges there are three layers. So you've got the dura mater, which is the outer layer that lines the skull and vertebral canal. You've got the arachnoid mater, which is the filamentous middle membrane that the CSF flows through. The pia mater, which is a thin membrane that lines the surface of the brain and the spinal cord. And then you've also got the subarachnoid space, which is the space between the arachnoid and the pia mater where the fluid flows through. Okay, CSF is produced in the choroid plexuses. About 20 mLs is produced from plasma every hour, and a volume of 90 to 150 mLs is maintained in adults. It's closer to 10, the range is 10 to 60 mLs in neonates. The choroid plexuses are a network of capillaries that forms the CSF by selective filtration. So the chemical composition of the CSF does not resemble an ultrafiltrate of plasma. Endothelial cells in the choroid plexuses are very tightly woven, and they form what is known as the blood-brain barrier, which protects the brain from chemicals and other substances circulating in the brain that could harm the brain. Disruption of that blood-brain barrier by diseases such as meningitis, multiple sclerosis, can allow white cells, proteins, and other chemicals to enter the CSF. Here's just a visual of the different areas of the brain, and you can see where the CSF is formed. Okay, specimen handling and collection. So it's collected via a process called lumbar puncture. You've probably heard it referred to as a spinal tap. And care has to be taken to prevent infection and neural tissue damage during that process. So it has to have to maintain a sterile environment. The volume that's removed is based on the available volume from the patient. So that one consideration could be, is the patient an adult versus a child? And it also is based on the opening pressure of the CSF as the needle first enters the subarachnoid space. Specimens are collected in 3 or 4 sterile tubes, and that is dependent on the lab that you work in, just whichever procedure they decide to use, whether they want to use a 3-tube collection or a 4-tube collection. And those tubes are labeled in the order in which they are collected. So and I've got a example here of how those tubes would be distributed to different parts of the lab. So tube 1 would be used for chemical and serological tests, because it's going to be less affected by, or those tests are going to be less affected by, blood or bacteria that might have been introduced into the sample as a result of the TAP procedure. Tube 2 would be used for microbiology testing. Tube 3 is going to be used for the cell count in a 3-tube collection, because it's least likely to contain cells introduced during the TAP. It's used just as an extra tube for additional testing in a 4-tube collection. And then tube 4 is used for the cell count in a 4-tube collection. Sometimes it might be drawn for micro testing to better exclude skin contamination. It can also be used for additional serological testing. And the reason why they want the cell count done on that last tube is just to ensure that any cells present are not the result of a traumatic tap. Of course, you always want to wear gloves when handling a spinal fluid, because meningitis can be very contagious. Any extra fluid that is left over after testing is complete should be frozen. And then here's that slide showing the 4-tube collection versus the 3. A normal CSF is clear and colorless, so really it looks like water, but it can also be described as cloudy or turbid, milky, xanthochromic, hemolyzed, or bloody. So xanthochromia is a term used to describe CSF that is yellow, pink, or orange, and that color is due to red cell degradation products. Other causes could be increased serum bilirubin, presence of the pigment carotene, increased protein concentrations, and melanoma pigment. Cloudy or turbid, cloudy, turbid, or milky can be due to increased proteins or lipids in the fluid. It could also be indicative of an infection. Okay, a traumatic tap. So if you receive a bloody CSF in the lab, it could be due, that blood in the sample could be due to an actual intracranial hemorrhage or bleeding in the brain, or it could be due to what is referred to as a traumatic tap, which happens when the blood vessels are punctured during the spinal tap procedure. So how can you tell which is which? So uneven blood distribution is one way. So in a true cerebral hemorrhage, the amount of blood present in the tubes will be the same in all 3 tubes, whereas in a traumatic tap, the amount of blood present will decrease from the first tube drawn to the last tube. Clot formation. So a traumatic tap allows plasma fibrinogen to be introduced into the sample, and so the sample will be able to form a clot. In a true cerebral hemorrhage, there is not enough fibrinogen present for the specimen to clot. You could do a D-dimer on the specimen to determine, but that type of testing on CSF is not common. There are also certain conditions in which damage to the blood-brain barrier allows increased protein and coag factors to be in the sample, but these conditions would not normally produce a bloody specimen. Xanthochromic supernatant. So xanthochromia represents blood that has been in the CSF for an extended period of time, so longer than the amount of time that it would take from a traumatic tap. However, a very recent hemorrhage could still produce a clear sample, so there is other testing that can be performed to interpret the results. Okay, so normally the white cell count is going to be done on the CSF to determine the presence of an infection. Red cell counts are done to determine if there has been a traumatic tap. So a lot of times physicians will request that the cell counts be done on the first and the last tubes drawn so that they can compare them to one another. So that's how they're going to determine if it's a traumatic tap or not. Cell counts should be done within the first hour after receiving the sample, because cells can begin to lyse within the first hour after collection. Okay, so in adults, normal CSF can contain 0 to 5 white cells per microliter. In children, a CSF can contain up to 30 mononuclear cells per microliter. Specimens can contain up to 200 white cells or 400 red cells per microliter, and the sample still be clear, so it's important that the samples be examined microscopically. Automated cell counts are available on some instruments. The cell counts are more commonly done on a hemocytometer, though, or it's also referred to as a Neubauer counting chamber. So if you have a hematology analyzer that has been validated to perform testing on spinal fluid, then by all means use it if you can. However, most of the time the cell counts are going to be so low in spinal fluid that the counts usually do not meet the linearity parameters for the instrument. So usually you're not able to run that sample on the instrument. Usually you're going to be counting them on the hemocytometer anyway. A grossly bloody or cloudy specimen may require dilution. If that's the case, you can use saline. Okay, and the standard Neubauer formula for determining your cell count. So we've got the average number of cells. So the chamber, the hemocytometer, has 2 different sides to it. Let me switch over to this slide real quick. So this is kind of a top looking down image of a Neubauer counting chamber. So it's got 2 sides to it. So there's side 1 that we would count, there's side 2. So you're counting each side, getting the average number of cells, multiply that times the dilution if you used one, divided by the number of squares counted on one side times the volume of each side, or of each square, sorry, which is 0.1. Okay, so we've got an example here. So if you counted 30 white cells on side 1, you counted 42 white cells on side 2, you have to get the average of that. So 42 plus 30 is 72, divided by 2 is 36. You've used a dilution of 1 to 6. The 4 large and 1 center white cell squares were counted. So let me flip over to this real quick. So this is what you're looking at when you're looking into the microscope, into the Neubauer counting chamber. So the big ones are considered the white cell counting chamber. So we've counted the 4 corner, so 1, 3, 7, and 9, and the center, 5. So those are the squares that have been counted. Okay, so we got our average, which was 36, multiplied it times 6 because that's our dilution. We counted 5 squares on each side times 0.1, so that was the volume of the square. So 36 times 6 is 216, 5 times 0.1 is 0.5. So when you divide 216 by 0.5, you come up with a result of 432 cells per microliter. Okay, there is also a modified formula. Let me back up just a second. This formula can be used whether the sample has been diluted or not. And you know, if the sample was not diluted, then you would just use a 1 in the place of that 6 right there. There is also a modified formula that can be used, which counts the same squares each time. So in the formula that we just looked at, that one has a little bit of flexibility to it because you can use it whether the sample's been diluted or not. The number and size of the squares can be varied. So in this modified formula, it's kind of like if certain conditions are met, you can use this modified formula. So it counts the same squares each time. So that's one of the criteria. And when using this formula, there's no need to correct for volume counted as long as the same squares are counted. So in this modified formula, it uses the 4 corner and the 1 center square of the white cell counting chamber. So again, it's the picture that we just looked at. It's the same squares. Just as a note, acetic acid can be used to lyse red cells in order to get a more accurate white cell count, but you really wouldn't want to do that unless it's absolutely necessary, because most of the time the volume of sample that you receive is not a lot. So you would definitely want to do that on an aliquot, you know, and not the entire tube. So you just have to make sure that you've got enough volume of sample to do that. Okay, so the modified formula counts 5 large squares on each side of the chamber, so that's your 10 squares. So 10 squares times that volume of the chamber is 1 microliter. So we're taking that into account with this modified formula. So in the modified formula, you're counting, it's the total number of cells counted, so not the average, times the dilution, times 1 microliter. So we've already taken that into account and plugged that into the formula there. Okay, so an example from the modified formula. So if you've got 327 red cells counted on side 1, 386 red cells on side 2, the dilution that you used was a 1 to 10. What's that red cell count? So again, there's our formula again. So 327 plus 386 is 713, so it's the total number of cells, times 10, because that was the dilution, times 1 microliter, so that accounts for the volume of the squares. gives us a result of 7,130 red cells. And again, here's that picture. This is the side image of a hemocytometer, and the one that's in this picture is not a disposable one, because you can tell from the side view here there is a cover slip that you have to add. So you load the sample on each side, which flushes it into that little chamber, and then you have to place the cover slip over top. But they do make disposable hemocytometers as well. And again, this is what you're looking at. So that's just a blown up version of this little square here. So that's what you're looking at. This is just kind of showing you the volume of each one of those chambers and the dimensions. This is kind of a neat visual here, just to show you the difference in size of different elements that you could see under the microscope when you're looking in that hemocytometer. Okay, for the differential. So usually a differential is going to be performed if the white cell count is greater than 10. And I know in my lab, if the white cell count is less than 10 and a differential has been ordered, we can cancel that differential as not indicated. Just because, I mean, think about how many, first of all, if there's not that many white cells, then that's not really an indication of infection. But think about how long it would take you to count a differential on a sample that only has a white count of 10 or less. You would prepare to perform the differential. You're going to prepare a cytospun slide and stain it with right stains, and then you count the 100 cells. And here's a picture of that cytocentrifuge. So, and there's a couple different, like, I guess, manufacturers that make these slide holder contraptions here. So this is a picture of the cytocentrifuge. This is like a microscopic holding contraption that is made to fit down in the little holes in the cytocentrifuge. So this is the back side of that contraption. Then you place the microscopic slide into it, and then there's this other like disposable chamber that goes onto the front of the slide, and all three of these pieces are connected. And you drop your sample into this little kind of filter here, or funnel, and you can see there's like a little tunnel that leads to the back of the slide. So it's open in the back, and that concentrates it as it's centrifuging. It pushes that sample through that little tunnel and concentrates it into one little area on the slide. So this is what you're left with. And the reason why we do that is because usually if there are elements in the spinal fluid, or really any body fluid, there may not be many. So if there are any, we want to increase our chances of recovering or seeing those elements. So that's why we do the cytospun slide. Okay, so lymphs and monos are going to be the main white cells seen in CSF, not as many neutrophils. Generally, there will be a higher lymph to mono ratio seen in adults. It's usually 70-30, and the ratio is usually the opposite in children. A predominance of neutrophils is indicative of bacterial meningitis, whereas predominance of lymphs and or monos could suggest meningitis of viral, tubercular, fungal, or parasitic origin. Other cells that can be seen in CSF include immature leukocytes, eosinophils, plasma cells, macrophages, increased tissue cells, and malignant cells. If NRBCs are found, this indicates contamination with bone marrow during the tap. Okay, so the significance of different cells in the CSF. So again, lymphs or monos are going to be common in viral, tubercular, and fungal meningitis. Reactive lymphs are common in viral infections. Increased lymphs are seen in cases of HIV and AIDS. and a moderately elevated white cell count in conjunction with increased normal and reactive lymphs can be an indication of multiple sclerosis or some other type of degenerative neurological disease. Eosinophils are associated with parasitic infections, fungal infections, especially Coccidioides immitis, and introduction of foreign material such as medications or shunts. Macrophages, so their purpose in our body is to remove cellular debris and foreign objects. They can be seen following repeated taps or a previous hemorrhage. They can look like a mono, but they're going to have more cytoplasm. Some non-pathologically significant cells usually are going to be seen after diagnostic procedures. A lot of times you'll see them appear in clusters. They'll have a uniform appearance. They come from the epithelial lining of the of different parts of the brain. Malignant cells of hematologic origin could be lymphoblasts, myeloblasts, monoblasts, which can be seen in leukemia. And then other malignant cells, non-hematological origin, would be tumor cells. Okay, the chemistry testing that we can perform. So reference values for CSF chemicals will not be the same as for the chemicals in plasma because of that selective filtration that occurs in the brain. So protein, normal CSF only contains a very small amount of protein. The reference range is 15 to 45 milligrams per deciliter, so not grams per deciliter as you would see in plasma values, but that can also vary some depending on the lab and the method used. Higher values can be seen in infants and in people over 40 years old. The significance of protein in CSF, so if it's elevated, this would indicate damage to the blood-brain barrier. The most common cause is going to be due to meningitis or hemorrhage. Immunoglobulin production within the CSF, decreased normal protein clearance from the fluid, and neural tissue degeneration. Decreased protein values could indicate leaking fluid is leaking from the central nervous system. Okay, electrophoresis can be used to differentiate the types of proteins that are found in CSF. So this can help to diagnose inflammations, leukemia, lymphoma, viral infections, HIV, MS, neurosyphilis, Guillain-Barré syndrome. Most of the time the CSF would be compared to the serum electrophoresis result for those determinations. So usually when when a CSF is ordered, when they're performing the spinal tap, they're going to draw a tube of blood at the same time. Okay, the glucose, CSF glucose values will be 60 to 70% of the plasma glucose value. So you would need to compare it to a blood sample for accurate determination. Significance of the glucose in CSF, so if it's elevated, this is always as a result of elevated blood glucose. If it's decreased, so this has diagnostic value in determining the cause of meningitis. So if it's markedly decreased with increased white cell counts with a predominance of neutrophils, this indicates bacterial meningitis. If the white cells are lymphs, tubercular meningitis is suspected. If you've got a normal glucose with increased lymphs, then viral is more likely. Okay, the lactate can help differentiate between fungal, bacterial, and tubercular meningitis. So if the result is greater than 35 milligrams per deciliter, that would indicate bacterial. If it's less than 25 milligrams per deciliter, that would indicate viral. If it's between the two numbers, that could indicate tubercular meningitis. Okay, then the glutamine. Glutamine is a waste product of ammonia, and you'll have elevated levels seen in liver disorders that will result in increased blood and CSF ammonia levels. So glutamine gives an indirect measurement of ammonia while being more stable. If the levels get high enough, this can lead to coma. Okay, microbiology testing. So the purpose of micro testing on CSF is to identify the causative agent of meningitis. So the gram stain can provide that preliminary diagnosis. And just, if there are bacteria present in the sample, then it's a sterile body fluid. So we're going to call that as a critical result. Most labs require that those gram stain results be out within a certain amount of time, just because of the the possibility that the patient is undergoing something life-threatening. Cultures can take anywhere from 24 hours to 6 weeks, depending on what the causative agent of meningitis is. So if it's tubercular meningitis, that organism can take up to 6 weeks to start to grow. So in addition to cultures, other tests are also available. So you can do the acid-fast stain, India ink preparation. There are latex agglutination tests available to identify different organisms. Okay, so the gram stain is used for detection of bacterial and fungal organisms. Again, the sample has to be cytocentrifuged. It can be hard to interpret because normally there's not going to be anything in the CSF, so it is really hard to find anything to focus on when you're looking at the slide. And also organisms are going to be fewer at the onset of the disease. Usually you'll have at least maybe like a red cell that can help you get initially focused on that slide. False positives are common because of debris that can be mistaken for organisms. And then some of the more commonly isolated organisms as the causative agent of meningitis, Strep pneumoniae, Haemophilus influenza, E. coli, Neisseria meningitidis, Listeria monocytogenes, Strep agalactiae, and Naegleria fowleri. Okay, sorry, let me back up a second. So there's also, like I said, acid-fast stains would be performed if tubercular meningitis is suspected. So with the organism Mycobacterium tuberculosis, that positive report from the acid-fast stain is extremely valuable just because those cultures for Mycobacterium take so long. India ink prep would be done when fungal meningitis is suspected, often in conjunction with a gram stain, so that it's used for identification of Cryptococcus neoformans, which is a frequently encountered complication of AIDS. And it produces what they refer to as a starburst pattern on the gram stain, which can be seen more often than positive India ink. And again, there are latex agglutination tests that are available that are more sensitive for those organisms. They make latex agglutination testing kits available to detect group B strep, Haemophilus, strep pneumo, Neisseria, and E. coli. And then finally, serologic testing can be used for the detection of neurosyphilis caused by the organism Treponema pallidum. So those serologic tests are testing for antibodies. The CDC recommended test is the VDRL, though it's not as sensitive as FTA-ABS. So you can really use the RPR or RPR stands for rapid plasma reagin. It's not as recommended, but I know a lot of labs still use it. So the RPR or the VDRL would be more like screening tests, and then FTA ABS, and then there's another one now called TPPA. Either one of those could be used for confirmation testing. Okay, this next little unit is on, at the beginning of the lecture I said synovial fluid, but it's on semen analysis. So we're going to talk a little bit about this. Okay, so we do semen analysis for the purposes of either fertility testing, so couples that may be having trouble getting pregnant and they want to try to find out the reason why, or we do it for post-vasectomy analysis. So semen consists of spermatozoa, seminal fluid, prostate fluid, and fluid from the bulbourethral glands. A complete collection is essential because all the fractions in the seminal fluid need to be present for accurate analysis. So the patient's going to be given detailed instructions on how to collect that specimen. Okay, so abstinence, we want the patient to have at least 2 days, but no more than 7 days. Somewhere around 2 to 3 days is recommended. Specimens should be kept at room temperature or body temperature until it can be delivered to the lab. It should be delivered within 1 hour of collection. The time of collection and the time of receipt into the lab should be recorded. And no lubrication should be used. Anti-spermicidal condoms if the sample is collected by intercourse. Though masturbation is the preferred method of collection because sperm could be lost during intercourse because the majority of sperm are present in the first part of the ejaculate, and the acidic pH of the vagina could affect the results. And usually when we have a patient who has to collect a semen sample, they're given kind of a piece of paper that has all of these instructions on it, but there's also information that they have to fill out. So we ask for, the patient's name and date of birth, of course, their partner's name, the method that it was collected. They have to write down the time that it was collected. We note the time that it's received into the lab. So, and all of this is because those different variables could affect the results of the testing. Okay, a seminal fluid obviously is a potential reservoir for HIV and hepatitis, so you're going to use standard precautions, and then the sample should be discarded as biohazardous waste. Normal semen has a gray to white color and will be translucent with a musty odor. It can appear almost clear when the sperm concentrations are very low. White and turbid samples could contain white cells and could indicate infection. A reddish color could indicate red cells, but that's abnormal. Semen with a yellowish color could be either contaminated with urine or could be collection following prolonged abstinence or certain medications. Urine is toxic to sperm and can affect the portion of the evaluation called the motility evaluation. Okay, liquefaction. So a fresh sample is clotted, but it should liquefy within 30 to 60 minutes after collection. If the sample is not liquefied after 60 minutes, this should be noted on the report because it could indicate a deficiency in prostatic enzymes, which are essential for the liquefaction and coagulation of semen. Normal volume would be between 2 to 5 mLs. Increased volumes can be seen after periods of extended abstinence. Decreased volume is associated with infertility or could be also just due to an incomplete specimen collection. Viscosity refers to the consistency of the fluid. It's that term or that concept is somewhat related to liquefaction. If the specimen does not liquefy, then it will be reported to have increased viscosity. And the viscosity is tested by drawing the sample into a pipette, and you should be able to form distinct drops, not be mucousy. Okay, pH should be measured within 1 hour after collection, and the normal range is usually 7.2 to 8.0. Increased pH could indicate possible infection within the reproductive tract. A decreased pH could be due to increased prostatic fluid, an ejaculatory duct obstruction, poorly developed seminal vesicles, and that test can be performed using the pH pad of your urine reagent strip, or your lab may also have dedicated pH paper. Okay, so concentration and sperm count. So fertilization occurs with 1 sperm, but the actual number of sperm can be an indication of fertility. So factors that can affect sperm concentrations and count include the days of abstinence, infection, stress. The concentration is considered the number of sperm per milliliter, whereas the count is the number of sperm per ejaculate. So normal concentration is 20 to 250 sperm per milliliter, and normal counts, which can be calculated as the concentration times the volume, count of greater than 40 million is considered normal. The count is performed using your hemocytometer, and the fluid has to be diluted. So it's done with a 1 to 20 dilution, and there's a special fluid, seminal diluting fluid, that you would use for that purpose. The diluting fluid immobilizes the sperm so that they can be counted. So again, you're going to count both sides of those, the hemocytometer, the 4 outer and 1 inner square of the red cell counting area is where you're going to count. The counts must match within 10% on each side. So remember, you're counting both sides of the hemocytometer, so those counts that you come up with should match within 10% of each other. When these squares are counted, the average number of sperm counted can be multiplied by 1 million to determine the sperm concentration per milliliter. So again, if you count those specific squares on the hemocytometer, you can get that average number, multiply times 1 million, and there's your count. The term azoospermic means that no sperm at all are present. Okay, and then here is again a picture of that, what you're looking at when you're looking down into the microscope at the hemocytometer. And for the sperm count, you're counting those 4 corner and 1 center square of the red cell counting chamber. Okay, motility. Sperm should demonstrate what's referred to as forward progression, and this gets reported as a percentage that is estimated just by visual assessment. Should be done at 30 minutes and at 3 hours. So you'll have 2 different motility results. Then the percentage reported represents the number of sperm that are demonstrating that forward progression. So, and it's performed just by putting a drop of the sample on a slide and adding a cover slip, and then you view it under the microscope. Morphology. So sperm that are morphologically abnormal can cause issues with infertility. So this would be a parameter that's evaluated by the pathologist in regards to the structure of the head, neck piece, mid piece, and tail. So this is not something that the tech would do. It's evaluated by preparing a slide in the same way that you would a peripheral blood smear, and then you have to fix it with a spray fixative, and then it gets stained and observed by the pathologist. Some additional testing that can be done. So additional tests may be ordered if there are any abnormal findings in the routine semen analysis. One of those could be vitality, which may be ordered when the concentration is normal, but motility is decreased. Fructose could be ordered with low sperm concentrations, which may be caused by abnormalities of the seminal vesicles or vas deferens. Anti-sperm antibodies. So this is kind of an interesting concept. So they can be present in men and women, though they're more common in men, and it can be a possible cause of infertility. It can be detected in semen, cervical mucosa, and serum. So normally semen would not be encountered by the male immune system, but it can be as a result of infection, vasectomy, some type of trauma. So if it does, then the body will see it as foreign. It can produce antibodies, and so the body will try to fight it off, causing the sperm to be damaged. When the female partner comes in contact with the damaged spermatozoa, then she can begin to produce antibodies. Antibodies could be suspected in the male if clumps are seen during the semen analysis. They can be suspected in females if the semen analysis is normal but continued infertility is a problem. And this can be tested for in women by mixing semen with the female's serum or cervical mucosa and looking for agglutination. They have quick kits also available for antibody detection. Okay, microbial testing. So when white cells are present, they can be an indication of an infection. So sometimes cultures are requested for detection of chlamydia trachomatis, mycoplasma hominis, ureaplasma ureolyticum. Chemistry testing, just be aware that there is chemistry testing available to detect enzymes and other elements in the semen, which can indicate reasons for infertility or other problems. Semen detection. So this could be used in alleged rape cases. So in this case, you're just looking for the presence of sperm. And then post-vasectomy testing, again, you're only looking for the presence or absence of sperm. So, and several specimens should be tested at certain intervals for a certain period of time following the vasectomy. And the patient should have 2 consecutive evaluations where no sperm are seen. This specimen should be centrifuged before examining. And again, that's just so that if there are any present, which presumably if the patient's had a vasectomy, there should not be any sperm, but if there are any left, they would be present in very, very low numbers. So you want to increase your chances of seeing them if they are present. So you want to centrifuge. And again, post vasectomy, all you're reporting out is are there sperm present or not? And even if you see 1 sperm, you have to report that out. Okay, so that is the end of this lecture for Unit 4. In the next unit is when we'll be picking up with synovial fluid and I think the other serous fluids.